Thank you very much, Masum. So first, I want to introduce to all today's presenter is Dr. Masum Rahman. He is graduated from Raghib Rabea Medical College, and after that, he wanted to pursue uh, his one of the biggest challenge to be a neurosurgeon in US. So for that reason, he came to do some research work in 2018, I guess. So from now, he is a very successful researcher. So during these 4 years, he published so many papers. So this is one of his original research paper. So today he will present this paper. So I would like to thanks to everyone for joining this journal club. And now I'd like to ask Masum Rahman to start his presentation. Thank you very much.
All right. Assalamu alaikum. Thanks everyone for joining us. Today I'm going to present one of my work that I'm very proud of, and I'm very grateful to my team to be part of it. That is selective vulnerability of the senescent glioblastoma cells. There is nothing to disclose about this study, but there is one thing we need to disclose, that we are going to record this whole session and we may have publish it in the public platforms. All right. So in today's presentation, at first we will talk about what is the problem and what we hypothesized about to solve this problem, and how we conducted this study and what is our conclusion. So the biggest problem is in this study we studied the glioblastoma, which is the most common primary malignant brain tumor. The biggest problem with this tumor is still the survival is very low, only 15 months of median survival. Then what's going on actually here? The current standard care of this therapy is, after a patient diagnosed with glioblastoma, we do surgical resection. After that, patient undergoes chemoradiation therapy, but eventually in every patient develop the recurrent tumor. So if you see the right side of the scan, MRI scan, the far left scan is showing the primary diagnosis of the glioblastoma, and the middle picture showing that we took this tumor out, but unfortunately this tumor is very, very infiltrative. So whatever you do, it's impossible to take all of the tumor cells out of this brain. That's why we have to radiate the brain and provide some chemotherapeutics to stop the tumor cell growth. But what's the wrong? Like, why after 15 months or after 9 months tumors comes back? That means in these 18 months, tumor cells are not dividing, they're not proliferating, but eventually they escape their non-proliferative state and start dividing. So what is the problem here? The problem is the recurrence. And if we look deep into the problem, what's going on here? So what we see, there is a latent period of time when in this time, this disease is very stable. The tumor is not growing, the tumor cells are not dividing, but after that, they start eventually growing. So there is a term we call in this period is latent period, or latent glioma, or the pre-recurrent glioma. So this is the terminology, and this is the novelty of this study. Like, what is the novelty? So the novelty is this is the first study have been done in this pre-recurrent stage of the glioma, and this is the first study when we studied senolytic drug, a different kind of drug that's I'm going to talk about it today in the treatment of glioblastoma. So to target this latent glioma or pre-recurrent stage of glioma, how we form our hypothesis. So our hypothesis is, in this pre-recurrent stage, due to this chemoradiation, the residual tumor cells stop dividing. That means the chemoradiated residual tumor cells don't divide for a period of time. And we define this state as senescence. So we know that senescence is a state of cell when cell stop dividing, but at the same time, they have some mechanism to survive. And our tumor cell don't follow the textbook rules. So eventually, this cell escape from the senescent state and start reproliferating, and we demonstrate the recurrence in the patient. So this is the problem. So our central hypothesis is this pre-recurrent glioma is the one which is the residual tumor cells, got chemoradiation therapy, and they are equivalent to the senescent glioma. As they are senescent cell, these residual glioma cells are senescent cell. So our hypothesis is senolytic drug might be able to eradicate this senescent residual glioma cell. That was the hypothesis. So again, I'm talking about, I was talking about the novelty of this study. The one novelty of this study is we studied in the pre-recurrent stage of the glioma. And second novelty of this study is using senolytic drug as a treatment of glioma. Now, for the first part of our hypothesis, we have to demonstrate that yes, the chemoradiation stopped the cell division in our model of the experiment. We used human-derived patient tumor from the Mayo Clinic biobank of the glioblastoma. We radiated a different dose, and we demonstrated that yes, after chemoradiation, cell stop dividing. So if you look at this first graph, the first graph indicates the x-axis is the post-radiation days, and y-axis indicates the cell proliferation status. That's called confluence. Confluence when I plate cells in a dish, in a tissue culture dish. So if the cell don't grow, that means the confluence remain the same. It's not to become overly crowded. So if you look at the dose after above certain level of dose, the cell stop dividing. That support our hypothesis that radiation stop the cell division, and we can see the senescence. The next step is this cell division are relevant to the senescence. That's why we used a qualitative biomarker that is called qualitative marker that is called beta-gal staining. On the bottom of my slide, this is the staining of beta-gal staining. So senescence is a character that develops over time. It takes time to develop. So so those cells are radiated cells. And eventually we see at the day zero, that means almost equal to the non-radiated cell. And over the period of time, the cells become blue. So blue means presence of senescent cells. So as the cells turn into blue, that means those cells are senescent. So the two things, the cell stop dividing, and this stoppage of division due to senescence. We get this concept from this slide. The next. In this slide, I'm trying to educate ourselves that what we are going to look at it. At this phase, we need to find a drug that selectively kill our senescent glioblastoma cells. So what we did an in vitro drug screening test with different targets of senolytics, different candidates of senolytic drugs. Now I'm going to say that like what we expected to see. So if you look at the drug A, that means, so here two groups of cell, non-radiated means proliferating non-senescent cell, and 15 gray radiated cell means non-proliferating senescent cells. So drug A kill all of the cells regardless of the radiation status or the proliferation status. If we look at the drug B, that indicates this drug B selectively killed the senescent cells. That means drug B can be a senolytic drug, not the drug A. So we are looking for a drug is equivalent to the drug B. So in my upcoming slide, our goal is to find out that which is our drug B, okay? All right, so let's find the candidate drug. So our candidate drug is the senolytic drugs, right? So senolytic drugs has a different class. At this moment, it's very visitable. You don't have to look at it. Just we will use this slide after looking at the data that which drug act like drug B as a senolytic drug. Then we come to this table and we'll find out that which target we're looking for. Okay, so if we look at this data right now, I'm taking a moment to for the viewers to understand and to find out the candidate drug which looks like drug B, which drug selectively killed the radiated cells. So in this slide, my black data points, those are the non-radiated cells, and the purple data points, those are the radiated or senescent cells. So if we look at these two drugs, navitoclax and A133, these two drugs selectively killed the radiated cells, but not the non-radiated cells. That means these drugs selectively eliminate the senescent glioma, selectively killed the senescent glioma. If you look at this 100 nanomolar drug concentration, almost 50% of the 50% of the senescent cells gone, but about only 10% maybe the proliferating cells are gone. So we can tell this navitoclax and A133 could kill the senescent glioma cells. Let's look what is the mechanism of these two drugs. Okay, so here is the thing. Navitoclax is a dual targeting drug. It targets BCL2 and BCL-XL. What are these? These are the two anti-apoptotic proteins. And what are these target pathways? These target pathways are actually the anti-apoptotic pathways. So let's remember what is the meaning of senescent cells. Senescent cells, these cells are not dividing, but they are remaining in a certain state. So they remain, they survive after the chemoradiation. How they survive? How do they survive? So they are surviving by increasing some anti-apoptotic pathway. And these anti-apoptotic pathways are the potential target for senolytic drugs. So what we tested all of these pathways with all of the candidate drugs, and we found there's overlapping of two anti-apoptotic pathway, that is BCL2 and BCL-XL. So the next step is to figure out actually which is the target, BCL2 or BCL-XL. All right, so for this, we included more cells and we included more drugs. So what we did, we included another BCL-XL inhibitor, that is A1155, and we added another selective BCL2 blockade, BCL2 inhibitor. That means if we do this screening, then we can clearly tell actually which is the target, BCL2 or BCL-XL. So let's start with the BCL2 venetoclax. If we look at the venetoclax, that does not look, these drugs could kill radiated senescent GBM selectively. We cannot tell that. But if we see that navitoclax, okay, it's doing that, but navitoclax is something target, and navitoclax is the drug which is targeting BCL-XL and also the BCL2. But if we look at the A133 and A1155, those are the selective BCL-XL inhibitor, and these BCL-XL inhibitors are selectively killing the senescent glioblastoma cells. So the conclusion from this slide is BCL-XL is the target protein, target pathway, not the BCL2. The next question remains. So although we included four different cell lines, still the question of reproducibility remains, that is this can be true for other glioma cell lines. So this is the reproducibility. We took several tumor cells, several human-derived patient glioma cells. This gave us an opportunity to test a diversity of molecular diversity and different status, for example, primary versus recurrent. And having all of this cell line, we tested, and we can tell that yes, this BCL-XL inhibitor could selectively kill the senescent glioma regardless of the molecular classification and regardless of the type, either primary or recurrent. The next question remains, if so we are hypothesizing that this senescent glioblastoma increasing the anti-apoptotic protein BCL-XL, and by inhibiting BCL-XL, we are selectively killing this senescent glioma. That's what we have been proposing till this slide. Now we should prove that we are targeting the actual protein. That's why in this slide we are showing the data. In this experiment, what we did, we selectively inhibit the protein in a method called knockdown, but we are not using the drug. So if we knock down the BCL-XL, then we can see the similar effect of what we have been seeing with the drug. So this is the mechanistic data that clearly demonstrated that yes, BCL-XL is the target. To compare with the other anti-apoptotic pathway, we also included the BCL-W and BCL-2, and the data showing, if we see the G, H, and I data, so these are the survival assay. So this is 15 gray, this is 0 gray, and this is the y-axis indicates the survival. So what we did BCL-XL knockdown here. So with BCL-XL knockdown, the 15 gray cell gone, almost died. And with this knockdown, proliferating 0 gray radiated cell did not die. If we compare with the BCL-W knockdown, so there is no difference between these two groups. If we look at BCL-2 knockdown, there is no difference between the proliferating GBM versus radiating senescent GBM. With this kind of experiment, at first we have to demonstrate that our knockdown worked very well. So this is the data we have shown that yes, the knockdown worked very, very well. That's why we can clearly tell them this by knocking down this BCL-XL, we could selectively kill the radiated glioblastoma cells. The next question remains, so we are inhibiting the anti-apoptotic proteins that results the cell death. So if we inhibit the anti-apoptotic pathway, that means the cell death should occur through the apoptosis, not other any mechanism like necrosis. That's why we have to demonstrate the increase of apoptotic activity in the senescent cell, senescent glioblastoma cell, when we target the BCL-XL protein. For that, you have to look at the data G. So here we have been saying that what we are expecting to see here, increased Caspase-3-7 activity. Increased Caspase-3-7 activity means increased apoptotic activity. So we blocked the BCL-XL in zero-grade radiated cell, and also 15, this is the with drug. So we have used this drug in zero-grade radiated cell, also in the 15-grade radiated cell. If you look at the data, the 15-grade radiated cell demonstrated much more increase in the Caspase-3-7 activity than the zero-grade radiated cell does. That means there are much more apoptotic activity in the radiated cells comparing to the proliferating cell. Well, what is the next question for translating this concept, translating this drug into the clinic? So into the clinic, the question remains, how soon after chemoradiation we can start this treatment in a patient? How soon after that we can do that? So that question means how soon after radiation this drug can be effective to eliminate senescent glioblastoma cells. For that, we did an experiment that is the time course, like how soon after radiation we could eliminate the cells. So if you look at this data, this time point actually important, but is still at any time point, this drug can selectively eliminate the radiated cells. That means the maturation of the senescent status is not required. Just after radiation, this drug can be effective to selectively kill the latent glioma cells. The next question remains, because we know in the patients there is another limitation called blood-brain barrier. So for that we need a certain dose exposure duration, how long we can maintain a therapeutic dose in the brain parenchyma. That's why we also tested in an experiment that is the duration important. Yes, the duration also important. And this drug also contradicts with the concept of senolytics in other study, where researcher reported that even a very short period of time, short duration of senolytic therapy can be helpful to eliminate senescent cells. But our data clearly contradicts with other, and it's conceivable because this is here we are dealing with tumor cells. These are very aggressive, and I like to thank my teammate, also my mentor, Dr. Terry Burns, and thanks everyone for joining us today. And I'm happy to receive any question if you have. And from this point, we can go to Bangladesh. I mean, combination of Bangla and question. We can ask the question in Bangla and answer in English or Bangla, whatever it is. Feel comfortable and feel free to ask. All right. Thank you, Moin bhai.
Yeah, thank you very much, Masum. So anyone have question, please unmute yourself and then ask.
Hello. Can you hear me?
Yes, we can.
Yes, my name is Dr. Pallab Sarkar. Hello, it's a wonderful presentation. To Masum bhai and everyone, I wanted to ask whether the drug, the BCL, which acts against BCL, will it's, as you said, it selectively targets irradiated cells. will it also target the normal healthy cell? Because BCL, I presume, is there in every cell in the body, right?
Yep.
So will it also have effect over there as well?
All right. So it's a fantastic question. To answer this question, at first, as our lab is a translation neuro-oncology lab, we at first we look at the safety and efficacy of the drug. This BCL-XL inhibitor have been using for the as a chemotherapeutic drug for hemat-oncology department for long. So yes, this anti-apoptotic BCL-XL protein is important for healthy normal cells. That's why what we want to see here a difference between the therapeutic dose and the toxic dose to the normal cell. To see this gap, we are demonstrating the data, the dose response curve, that yes, this drug can be toxic to the normal cell, but the concentration should be different. That is toxic concentration versus therapeutic concentration. These cells, the tumor cells, are much more dependent on the anti-apoptotic pathway. And when we hit them with the radiation and chemotherapeutics, they become more dependent on these anti-apoptotic pathways. So yes, this anti-apoptotic pathway is important for any cell we have, but these cells are very, very dependent on it, and we are taking the advantage of that. I hope I answer your question.
Yes. Thank you very much.
Actually my name is Mahfuz. I'm from UW Medicine. Really nice presentation. Appreciate.
Thank you.
I have a few questions. Like one question is, you always use your drug with radiation. Why don't you show any? Like I'm wonder, why don't you show any graph that shows the only drug, only radiation, drug plus radiation, both. So in that way, you can see the difference between the drug and radiation both.
Okay, so what is the next question?
So my question is like, is that for your drug, radiation is needed? Like, should the cells have the radiation and then your drug will work, or your drug will work any senescent cells?
Okay, so that's a fantastic question. I like to start sharing again to answer this question. Okay, so first of all, we decided to use senolytic drug to find our candidate drug for the glioblastoma, for the latent glioblastoma. Okay, for that, these drugs have been already demonstrated that they have senolytic property. But the main tricks of the senolytic drug is not all senolytics can kill all senescent cells. So they are very, very specific. For example, senescent cell in the lungs can sensitive to the one particular group of senolytic. Senescent cells in the brain can be sensitive to the one particular group of senolytics. So that's what we were looking for. Actually, which senolytic can be effective to eliminate the senescent glioma. So that was the first question. So the second portion of your question, that if this drug could eliminate the senescent cell from other part of the body or can kill other senescent cell, the answer is yes. They are well demonstrated and well described. Okay. Your first portion of the question is why we used after radiation, not before radiation, because this is the time frame we are looking for. So every patient gets surgery, every patient gets chemoradiation. So eventually after that, we have this problem, right? The disease is very stable, tumor is not growing. Then eventually after 10 months, tumor is coming back. So what's the problem in this time? So we are selectively looking for a group of cells, those don't remain silent forever. Those are restarting the cell proliferation. So we are particularly studying these group of cells, right? So these group of cells are very different than the proliferating cells. That's why we used in a sequence, not in conjunction. So that's the reason, because this is the time point. In patient, we do surgical resection first, then chemotherapeutics, then we wait to have the patient recurrence, then we treat the recurrent. What's wrong with this latent phase? What is the characteristic of these cells which is in this period of time? We are looking for these cells, and we need to address this cell first. Otherwise, yes, we can add treatment with radiation, but that does not answer our question. What if we found another chemotherapeutic that is a radiation sensitizer? Say we are getting another drug that is more effective when used in combination of radiation, right? Then it can be a new chemotherapeutic. It can be a new drug that can be used as radiosensitizer. Then we are following the same trend. Then we are making them senescent, and eventually, after a period of time, these senescent glioblastoma will certainly recur, right? So that means we are not addressing the problem at all. So our thing is, we can stop the cell proliferation in primary diagnosis. We can treat, but we cannot prevent them from the recurrence. So who is the responsible cell for recurrence? These latent cells. That's why we target to study these latent cells which occurs after chemoradiation. I think I answered your first portion of the question.
Yeah, I think so.
May I ask something?
Who?
My name is Ashish.
Sure.
May I? Yeah, okay. So I'm very much new about the cancer cell in the brain, but what I know about the cancer cell, that is the previous question Dr. Mahfouz asked you. So is that the drug for, let me clear one thing. Is that the drug for only post-operative or post-chemotherapeutic patient? Yes or no?
No, this is just a complete different class of drug. That that the primary classification of this drug are senolytic drug. So they have never been used in glioblastoma. So we are trying to introduce the concept of senolytic in the glioblastoma.
So this drug can be used from the beginning of the glioblastoma.
Well, so
this drug can be used after the surgery and chemotherapy. Then we will use for the stop of the recurrency.
Oh, okay. So having like, to answer this question, I have to repeat the same thing again. Like, if we end up, let me finish. If we end up finding another good drug as a chemotherapeutic drug, so at first you have to understand what is chemotherapeutic drug and what is senolytic drug. Chemotherapeutic drug can stop the cell proliferation. So after applying chemotherapeutic drug, some cancer cell died. Some cancer cell still remain. That's why we are observing the recurrence. If they could eliminate 100% of the cell, then fantastic. We did not see the recurrence. We don't have to see the recurrence, right? So if we end up discovering another potent chemotherapeutic drug, but it's impossible to eliminate 100% of the cell till now, maybe in near future it will be possible. So the chemotherapeutic drug has two effects. One, can kill cancer cells. Two, can produce senescent cancer cells. The senescent cancer cells not proliferate. That's why you are not having the tumor mass effect or any effect from the tumor. So this is the good thing about the chemotherapeutic drug. But what is the problem with this chemotherapeutic drug? Chemotherapeutic drugs did not solve our problem, because this senescent tumor cell eventually recur, eventually start proliferation after a certain period of time. That's why we are not looking for discovering a new chemotherapeutic drug that can be used during surgery or after surgery or with the radiation. We are looking for a solution that can we treat those senescent cells. Those are not dividing, because chemotherapeutic drug only work on dividing cells, only proliferating cells. So once those cells are not dividing, you cannot apply more radiation. You cannot apply more chemotherapeutics, right? Because you're done with the chemotherapeutic and radiation. Then how to deal with those latent cells that we know those cells will eventually produce tumor in future? That's why we introduced the concept of senolytic. Can we remove those latent cells? Those are already got chemoradiation. They're silent at this moment, but they will come back eventually. Can we eliminate those cells? So that was the goal of this study.
Okay, thank you. Perfect.
Yep.
Thank you so much.
Dr. Masum, I have one more question. Sorry for that many questions.
Yeah, sure.
Like we know that the 15K radiation is much higher.
Yeah.
So in the cell culture, you are working with the primary cell line, right? Is that cell line?
So these are the actually patient-derived cell line.
Right, primary cell line. Like kind of, so you collect the tumor and make the tumor, right?
Yep.
So yeah, so if you radiate the primary cell line, you will always get the like high number of cell death because you give the radiation, right?
Okay.
And my question is, how long, like when you add the drug after the radiation?
Okay, perfect.
Because you know that, as you say, after the radiation, the cell proliferation curve will drop, and then it may goes up.
Right. So okay. So I hear you. To find the time point, when should we treat the cell with the senolytic drug? At first, we have to look at our first data. Okay, let's look at our first data. In our first data, if you see, so let's pick one dose, the dose most frequently used in this study, that is 15 gray of radiation. So let's find the 15 gray radiation. So this is somewhere in this area. If you see, so why there is a gap here? Because we allowed the cell to be dead. So those are done in this time. Then we plated this cell. We kept our plate in the incus site and studied their proliferation status. If you look at these cells, this cell remains silent without proliferation till 40 days. And most of my experiment, except the last data point, was done around the day 20. That means in this day 20, you cannot have the acute effect of the radiation, because just after the radiation, we have this data also I have not shown here. We optimize that. Cell death can persist until 7 days or 5 days. So you will see the effect of radiation, that cell count are going down gradually till the 7 days or 5 days, depends on the cell line. But see, after the 10 days, it's almost stable. It's not going down further. So in all of my study, I choose the time point of day 20. In this day 20, we took equal number of senescent cells and equal number of proliferating glioblastoma cells and did our drug screening to see what is the effect. So did I answer your question? So yes, the question is fantastic. The time point is also important because we want to eliminate the effect of radiation on the cell death. We don't want to screw this up. And even in the acute phase of radiation, the BCL-XL, the anti-apoptotic pathways goes up. That does not describe about the latent glioma cell that can persist many years. And for the in vitro, I have not shown this data here, but it's in the supplemental or somewhere, I think. We have a pattern of, can we reproduce the recurrence model in the in vitro? Yes, we did. After 60 days or after 52 days, depends on the cell line, cells start dividing. So we reproduced that in the in vitro as well. So we did all of our drug screening in the stable phase of the radiated cell, when these cells are not dying, when these cells are not dividing. So they are particularly senescent glioma. That's why we did this study, this characterization first, that when to screen with senolytic drug.
Okay. So you also use some proliferative markers, like the cells growing after the.
Yeah, that's correct. So again, we are not showing all of the.
Yeah, I know. Yeah, it's not possible
right.
to show everything.
Yeah, right. So yes, we did that. We have PCR data. In PCR data, because PCR data helped us to characterize the GBM senescence, because in senescence you will see the rays of sus factor, number one. Number two, the proliferation. It's a good question also. We did our proliferation Ki67 marker. Is that Ki67 or 65?
67.
67. So we did our Ki67 staining, also Ki67 PCR, and we demonstrate that yes, the cells are not dividing at all.
So you are using also the InQSite, right?
Right. We are using InQSite for this study.
So this is the technical question actually. I'm also using the InQSite.
Perfect.
Yeah. So the cells you plate onto the InQSite, which plate did you use? Like, is that 6-well or?
No. So because I have been using a variety of radiation dose, that's why we
long time point,
right. That's why we ended up deciding that we are playing with the 96-well plate, and our InQSite can do anything technically. So any InQSite I think can read the 96-well plate. So we decided to do in the 96-well plate.
Okay.
And to have more data point, to have more statistical power also.
Okay. And did you use any dye or how can like InQSite has also some red and green fluorescent, and
right.
can use the bright field?
No, we just use the bright field and the confluency. And
okay.
we have been we optimized a little bit, because the real challenge is you have to radiate cells in one dish, then plate in equal number if you want to compare with the non-radiated cell. So that's why you have to radiate in one dish, then transfer to the actual 96-well plate for the InQSite study. The next question is, when you plate the cells in that 96-well plate, after trypsinization, cells shrink.
Right.
After a certain period, they start to like relax,
Right.
As we are using the confluency assay, this relaxation can mimic the increase in the confluency. So we also optimize that phase, that background, that no, we are not dealing with that relaxation to detect the false increase in the confluence in the radiated cell. So yeah, I think we are good enough with this. Yeah. Also this have been done in several cell lines.
Okay, good work.
Perfect, thank you.
Yeah, what is the future work for this future direction of this work?
Okay, so the future direction we have kind of, so we're still struggling to incorporate in the in vivo models. So in vivo models, these drugs are really hard to get into the brain. That's why we have been also using the convection enhanced delivery system in the brain. But it's really expensive. We are at this point we are looking for more grants, because if we need to do the convection CED deliveries, CED drug delivery, then we need more more fund. So our flank tumor model and IC tumor model, when we did not use the convection enhanced delivery, we did not see any difference. But the model where we could use the convection enhanced delivery, we did see the difference. So in vivo, it's very promising. So let's see where it goes.
Okay, so you are using the flank tumor model. What happened? Like, do you have any idea? Like, you can use the orthotopic model.
So orthotopic model means it can be flank, it can be IC, right?
Yeah.
Is there any difference? I don't think so.
I think orthotopic model is.
I think those are all very technical questions. So I'd like to invite Mahfouz to personally contact with Masum.
Sure, of course. I'm sorry.
Definitely. So you can contact with Mahfouz. I'll share his email address, and then
sure,
you can have all the technical question, ask him.
Yeah. Sure.
I'm very happy because we actually arranged this program for more collaboration. So you know
Right.
now, Mahfouz, that Masum is working like in these techniques and in this, he is expert. So you can also share your work and your experience with Masum. So this is the
Yeah.
actual purpose, to have everyone and share our work and collaboration. So thank you very much, Masum. So today, I think it's a successful presentation, and we wish you your success, and your journey will be more high and high up, and you can
Thank
cure
you.
glioblastoma patients.
One day. All right.
Definitely.
Thank you
So
so much, everyone.
that's how we would like to finish our presentation. So thank you very much. So
Bye bye.
Thank you.
bye bye.
Bye.
Thank you.